Welcome learners for another lectures on vaccines in biotechnology. Today we are going to deal with vector recombinant vaccines. The previous lectures we have seen vaccines and its types. So today we are going into the another area that is called vector recombinant vaccines. What is recombinant vector vaccines? So the recombinant vector vaccines are live replicating viruses. They are engineered to carry extra genes derived from a pathogen. That means these vectors will carry the foreign genes of their pathogen so that they can induce immune response based on the antigen present in their genome. So these foreign genes, they produce the proteins which are antigenic in nature. They will evoke immune response in the host cell, thereby giving protections. So the vector, in this case, we are using as a vaccine. So the recombinant vectors, that is a vector, mostly a virus, which is recombined with any foreign gene to make it as a vaccine. So examples for these recombinant vector vaccines. Viruses currently utilized as vaccine vectors. I have pointed out some examples here for you to understand about the different types of vaccines available in the market for recombinant vector vaccines. The first one is adenoviruses. That is, example is double-stranded DNA viruses. It is used in the recombinant vector vaccine. Other one is alpha viruses. That is single-stranded RNA viruses. Then pox viruses, is double-stranded DNA viruses. As well as we are having herpes viruses, which is double-stranded DNA viruses. So we can see here we have 4 different types of viruses used as vectors in vaccines. That is adenovirus, alpha virus, pox virus, and also herpes virus. So today we are going to see about the recombinant vector vaccine. For this we are taking an example for this virus is Vaccinia virus, comes under pox virus category. So we know that in the previous lectures that Edward Jenner, who is called father of vaccination, used this Vaccinia virus to vaccinate against smallpox, because this Vaccinia virus is a pox virus which mainly affects the cattle. So these Vaccinia virus, which is exposed to any human beings, they have the ability to withstand or can have the immune system to withstand against the smallpox so that it can be vaccinated indirectly. So this Vaccinia virus is a very good candidate for the live recombinant viral vaccine. So they use this Vaccinia virus for vaccination program. Why? Because for 3 important reasons we are using this virus. The first thing is it is a benign virus. It is not harmful. It is harmless virus. Another thing is that they can replicate in cytoplasm. So that is another advantage. It doesn't replicate in nucleus. It replicates in cytoplasm. Then likewise it is easy to store. These are the 3 important properties or the features which makes this virus a good candidate for vaccines. Now we will see about the Vaccinia virus. how it looks like and what is the structures looks like. This Vaccinia virus is a large complex enveloped virus which belongs to pox virus family. This virus have linear double-stranded DNA genome. Approximately, if you see, it is 190 kilobase of length of DNA is present in Vaccinia virus. That encodes approximately 250 genes. The dimension of this virion is roughly 360 into 270 into 250 nanometer. The dimension of this virus. So it is a double-stranded DNA virus, the Vaccinia virus. We should know about its mode of replication, how the virus replicates. Mainly the virus, Vaccinia, they replicate in host cytoplasm rather than in nucleus. And please remember that when it replicates in cytosol or cytoplasm, they should have all machineries to undergo this replication, transcription, everything there. All these machineries for transcription or replication process is present in this genome itself. This virus have all these things, so it provides all these things for the replication of the virus. So if you insert any foreign gene in the virus, they are under the control of the virus itself, and it will be expressed along the viral genome because the virus has the capacity for replication and transcription of its own, because it has all the potential for doing replication and transcription because of the presence of all factors it's having in its genome. So that if any foreign gene or genome or genes attached to this Vaccinia virus, they also replicated, transcribed along with viral genome because of the facilities present in this genome itself. The advantages of Vaccinia virus. Why we go for this Vaccinia virus? The main reasons why we choose this Vaccinia virus for recombinant vector vaccines is because I have told you in the previous slide, it is harmless, very easy to cultivate, and it is stable for years, and they stimulate both B and T cell lymphocytes. If you take subunit vaccines, they stimulate only B lymphocytes, whereas the vector recombinant vaccines stimulate both B and T cell. Likewise, these Vaccinia virus, that is vector-based vaccines, can vaccinate against different diseases at a single time. So these are the big advantages when we go for vector recombinant vaccines. Now we see about the production of this recombinant Vaccinia virus. It involves mainly two steps. What are they? That is, first, assembly of plasmid insertion vector. The second step is the production of recombinant Vaccinia viruses. These two are the important steps for the production of recombinant Vaccinia virus. The first step, that is assembly of plasmid insertion vector. First, what we do, we remove the DNA from the Vaccinia virus. By processing the virus, we take the DNA out of that virus. Then after taking the DNA of the virus, then we remove the genes, the specific genes from different pathogens, that is hepatitis B, herpes simplex virus, influenza. When we take the specific genes from these pathogenic viruses, then these genes will be inserted into the Vaccinia virus genome one by one. After inserting all these genes into the Vaccinia virus, then this Vaccinia virus gene is cloned in E. coli, that is to produce plasmid insertion vector. So what this plasmid insertion vector will have? It contains the foreign genes, that is the genes taken from hepatitis B, herpes and influenza. All these foreign genes will be having in this plasmid insertion vector, plus vaccinia genes and promoter genes. So this plasmid, which is present in the E. coli, which is grown in E. coli, will have viral genome, foreign genes and the promoter of the virus. So after the recombination occurs, this plasmid is removed from that E. coli, isolated and purified. See this diagram for your better understanding. We can see here the plasmid. Here if you see this plasmid, we can have the virus promoter, that is Vaccinia virus promoter. We have TK gene, that is thymidine kinase gene, and we have a cloned antigen gene, that is a foreign gene. So what happens here? The Vaccinia virus will have thymidine kinase gene. So this thymidine kinase gene, the homology of that, it is also present in the plasmid. To this plasmid, what happens? This plasmid contains the cloned gene and the promoter of the virus, plus the homology sequence for TK gene. So by this homology of the TK gene, there occurs recombination between the Vaccinia virus genome and the plasmid to form plasmid insertion vector. The second step is the production of recombinant Vaccinia virus. If you see here, we have the plasmid insertion vector, that is having the foreign genes along with the viral gene. So we take this plasmid insertion vector, recombinant plasmid insertion vector, we isolate and purify it. Then we take Vaccinia virus itself. These two we will infect the animal cells. So inside the animal cells, the viral, that is Vaccinia virus, replicates. So the plasmids are taken to produce recombinants, which I have seen in the previous slides. When the virus replicates, the plasmids and the viral genome, they will have the recombination because of the homology of the TK gene. That is, plasmid insertion vector incorporates its gene into the Vaccinia virus at the TK gene. TK stands for thymidine kinase gene, because this thymidine gene will have homology both in plasmid and also in the Vaccinia virus genome. There occurs the homology, there the recombination occurs between these two. So that what happens? The foreign gene present in the plasmid recombined with the viral genome, thereby the TK gene will be inactivated because of the insertion of the foreign gene from the plasmid. So these virus released from animal cells. So after this virus replicates and take this plasmid foreign gene into its genome, they become recombinant viral vaccines. And these viruses can be released from the animal cells and they can be used as vaccines. Diagram. We can see this a gene from the pathogen. We have a TK gene is inserted in between. So these is cloned with Vaccinia virus. Both will grow in the cells. There, this gene, the foreign pathogen gene, that is taken by the varicella virus, thereby it becomes recombinant, which can be purified and can be used as vaccines. Advantage of this, so authenticated antigens, they will closely resemble the natural antigens can be produced by these recombinant vector vaccines. Likewise, the virus can replicate in host cell, which enables amplification of antigens for the action of B and T cell lymphocytes. Likewise, these vector recombinant vaccines, they can give protection against several diseases with one recombinant vaccines. These are the advantage of recombinant vaccines, is that unknown risk of using this vaccine is prevailing. So only they use only for, not for human patients, only for animal disease. They are using these recombinant vector vaccines. Likewise, we cannot test these type of vaccines in the immunosuppressed individuals like AIDS patients because serious complication can occur. So these are the important thing about recombinant vector vaccines. So we will meet you again in another lecture. Thank you. Thank you very much.